Okay, so it's a pleasure to start a full session for virtual seminar in Precambrian geology. We have a full schedule, I think, until AGU, and Alex and I will put a schedule for 2025 until summer and start inviting people. So if you have suggestions, feel free to contact us or edit on the website. And today it's a pleasure to introduce Phoebe Cohen from Williams College. Phoebe graduated from Cornell University with a bachelor degree in Earth systems. And when she spent a couple of years as an assistant to a director in Paleontological Research Institute, after it, she did PhD at Harvard with Andy Knoll. And when she continued in MIT, working with NASA NOD until she started position in Williams College, where she is a full professor now. She already been a chair of department, and in addition, so her research mostly focused on biological evolution in late Precambrian, appearance of eukaryotes, and that's the topic she will present today. So it's all yours, Phoebe.
Thank you so much. And thanks to everyone for showing up to this first seminar of the fall. I'm going to be talking about new work that I've been doing in collaboration with a whole bunch of people who are on the next page, using single fossil organic carbon isotopes to better understand Proterozoic eukaryotic biology. So I'll be presenting two use cases using this technique in this talk today. Okay, so first of all, I would like to acknowledge the very many people who have contributed to this work, most principally Chris Junium at Syracuse University. This is very much a collaboration with Chris, who is the mad scientist behind the analytical tools that I'll be talking about today. This work is also was done in large part with collaborators who are former undergraduate students at Williams College, as well as other collaborators who provided insights into the ideas here, access to samples, and more. Okay, so this is a figure from a review paper that I wrote with Robin Codner a few years ago, where we attempted to basically take everything that we know about Proterozoic eukaryotic ecosystems and put them into one figure. And so what we're looking at here is the integration of the fossil record, organic biomarkers, molecular phylogenetics and phylogenomics, and redox proxies in a hand-wavy sort of way to look at the evolution, the tempo and mode of eukaryotic evolution in the Proterozoic. So the key features to see here, in my opinion, are that the molecular clock estimates for LECA, the last universal common ancestor of eukaryotes, range very broadly, which is what this sort of gray fuzzy bar represents here. But the presence of crown group red and green algae in the Archaeoplastida clade up here at about 1 billion years old, and the placement of that clade, Archaeoplastida, within the larger eukaryotic phylogeny tells us that crown group eukaryotes had to have evolved before 1 billion years ago. And then we see the subsequent radiation of eukaryotes, and the stars represent taxa that we think we can link to modern day groups, to crown groups. At the bottom here are just some of the sort of representative taxa, and you can see that, you know, we start getting more interesting things in the Tonian. And this is also where other work by Leanne Riedman, for example, has shown that there's an increase in diversity during this time period as well, leading up to the Snowball Earth events. The biomarker record, which I'll talk more about later, we start to see sterane eukaryotic biomarkers at around 750, 800 million years ago. The record before then is relatively spotty just because of access to appropriate material, but steranes, sort of crown group steranes, have not been detected. And we know that oxygen was changing during this time period with increasing surface oxygenation, but, you know, spatial and temporal variability in terms of euxinia and anoxia below the surface. So much of my work has focused on organic walled microfossils, which are mostly things like acritarchs, which are closed-walled organic microfossils of uncertain taxonomic affinity. They're very abundant and common through the Proterozoic and the Paleozoic as well, but they're poorly understood from almost every angle and are definitely polyphyletic in terms of being made by many different types of organisms. But most of them are understood to be somewhere within the eukaryotic clade because of their complexity and their size. And so I've spent a lot of my career trying to understand how we can use fossils like this to better understand Proterozoic ecosystems and evolution. And one of the things that has always been of interest to me is the fact that these fossils represent primary organic carbon created by an organism. And so I've always been interested in figuring out how we can use this primary organic carbon to understand early eukaryotes in the global carbon cycle. But because they're so small, until recently, analytical capabilities were just not able to analyze a single fossil. There's been some work using SIMS, for example, but there was no way of sort of doing sort of high throughput, high volume carbon isotope analyses on acritarchs. So that's where my collaboration with Chris comes in. Chris has developed a nano-EA modified elemental analyzer coupled to a stable isotope mass spec. And here's just a little cartoon of the system that involves a cryotrap, just showing you Chris's setup. And it allows for carbon isotopic analyses of around 10 nanomoles. We usually try to go for 20 to 100. And you can see that the reproducibility with reference material is less than about 0.5 per mil. Okay, so now we're gonna get into the use case of this approach, looking at Mesoproterozoic acritarchs. So coming back to this figure, one of the things that has long been a question is the disconnect between the record of crown group steranes and the fossil record itself. And so organic biomarkers suggest that eukaryotes don't contribute much to marine ecosystems until after 800 million years, but the fossil record and the phylogenetic tree tell a different story. And so why is there a disconnect here, and what can it tell us? And so to do this, we have to sort of take a little bit of a side step into understanding the relationship between eukaryotes and molecular oxygen. So here's my little cartoon of how we think eukaryotic evolution happened. So we have the last universal common ancestor of all living things, which diverges into bacteria and archaea at some point. And then we have Theca, the first eukaryotic common ancestor, which is essentially the cytoplasmic bag that will eventually turn into a crown group eukaryote. And this is an archaeal lineage, has the nucleocytoplasm that will ultimately become the host of the eukaryote. So now we maybe consider ourselves in the stem eukaryotes, depending on how you feel about that term. Okay, so at some point, oxygen rises a little bit, either globally or locally, and this lineage becomes oxygen tolerant. And that has to happen because the eukaryote, crown group eukaryotic clade, eventually has to host the mitochondrion, which uses oxygen, and so is thus itself oxygen tolerant. And we know that this lineage then evolves sterol synthesis, probably from horizontal gene transfer from bacteria. And around the same time, we don't know the order of events here, the lineage also acquires the mitochondrion via endosymbiosis and then becomes LECA, the last universal common ancestor. So there's this acquisition of mitochondrion and sterol synthesis, both of which require molecular oxygen, and thus an oxygen-tolerant cell. So this framework requires that sterols were being made relatively early, where we don't see them in the rock record. So LECA has a mitochondrion and can synthesize sterols, among other synapomorphies, shared derived traits. So if we have fossils that we think are post-LECA in the fossil record, then we should expect to see sterols in the rock record. But there have been many hypotheses to explain this disconnect, and just one of the people to propose this one is Susanna. So what if LECA evolved really late? Or what if eukaryotes weren't living in oxygenated environments and thus didn't need or produce sterols? So here's just a figure I took from one of Susanna's papers, where she sort of presents a framework for this hypothesis that many of the shared traits of eukaryotes might have evolved back here in the Mesoproterozoic, but sterol biosynthesis might not have evolved until later, once we sort of get to crown group eukaryotes. And there's lots of evidence from modern understanding of eukaryotes that would support this idea, this is a great quote from a Mueller paper that says the strictly aerobic lifestyle is one that arrived late on the scene of eukaryotic ecological specialization. And this figure is just showing the vast diversity of organelles in microeukaryotes in the modern that are not necessarily anaerobic. Okay, so one of the things that we have to think about is the fact that phylogeny tells us that we have major branches of the eukaryotes deep in the tree by 1 billion years ago. And it's possible that the organisms that produce eukaryotic fossils from before the late Mesoproterozoic didn't make sterols. But I have a hard time squaring the idea that none of them did until 800 million years ago, because it doesn't really make sense in a phylogenetic framework. There's definitely still a gap between the crown group fossil record and the sterane record. And that could be a rock record, sampling issue for sure. But there's also bacteria that can make sterols. And so the fact that we're not seeing any of them up until that point is still, I think, an important thing for us to investigate and think about. So recently, Brox and colleagues have presented this idea of protosterols and this sort of protosterol biota. So protosterols are molecules that have some, but not all of the traits of sort of a crown group sterol, and have been detected extensively in late Paleoproterozoic, Mesoproterozoic, and early, or sort of in some Tonian strata in the purple over here. And so the idea here is that maybe these protosterols are, basically the biomarker record of these stem group eukaryotes that we're just not seeing. There are still some problems with that, mainly that, oops, sorry, my video is messing up over here. Mainly that there's still a 200 to 300 million year old gap between crown group eukaryotic fossils and sterols. And this also assumes that the crown group didn't evolve until about 1 billion years ago, which is inconsistent with other lines of evidence. So even though the protosterol idea sort of adds some interesting sort of nuance or complexity and helps bridge some of these gaps, it doesn't entirely solve this problem. So to try to better understand where eukaryotes were living during the Mesoproterozoic and how they were living, we were able to obtain some material from the Altry 2 core of the Velkerri Formation in the Roper Group in the Northern Territory of Australia. And the age constraints on the Velkerri are about 1.4 billion years old. Many people have worked on this core, which is fabulous. And then so I'm showing some figures from one of Amber Jarrett's papers looking at various redox proxies through this core, which has been divided up into 3 members now, the Lower Kalala, the Amungi, and the Waiwori. Then these little cartoons from Jarrett 2019 showing sort of this conceptual models of the chemistry of the upper Altry and the middle. So in the middle Altry, we have more evidence of euxinia. We have elevated molybdenum and uranium proxies, and there's many other people, including many papers by Cox, who have also reproduced some of this work or done some of this work. And so we were able to obtain material through this core and try to look at what the fossils are telling us about the transition between these 2 members. There's actually no fossils in the lowest member, which is another interesting story about taphonomy, which I don't have time to get into today, but if folks have questions, I'm happy to talk about that. Okay, so what's the framework that we were using for this? So the biological pump in a well-mixed ocean, this is a little cartoon version. You have photosynthetic organisms that are preferentially uptaking C12, they sink and die. And in a well-mixed ocean, because of remineralization from respiration, that light CO2 then recirculates up to the surface ocean. And so we get a relatively shallow isotopic gradient. And I highly recommend Katja Meyer's 2016 paper, which does a beautiful job modeling this sort of scenario. But in a stratified ocean where we don't have as much mixing from the surface, that light carbon is essentially going to get trapped down below, and we'll end up with a much steeper gradient where there's a much more heavy enriched DIC signal at the surface and a sort of more efficient biological pump. So in the NSF grant that Chris and Susanna and I wrote many years ago now to fund this work, we presented these two alternative hypotheses. So the prevailing model is that early eukaryotes are restricted to oxic habitats. And in this situation, we would expect that eukaryotes would be living in an oxic water column, sort of all along this gradient, again, depending on water depth and photosynthetic versus heterotrophy, things like that. And in an anoxic ocean, they would only be living at the surface. And so in an anoxic stratified ocean, we would expect that the delta C13 of fossils would be heavier than bulk because they'd be recording the DIC either through photosynthesis or heterotrophy of this enriched surface water. Whereas in an alternative model where eukaryotes were living in anoxic parts of the ocean, we might expect something that looks like this, where we do have organisms incorporating this lighter DIC lower down in the water column, if we have sort of independent proxies telling us that there is anoxia at depth, and that we might see some fossils that are heavier, some fossils that are lighter, and some fossils that are equivalent in this sort of scenario. So we went ahead and tried to test this hypothesis using fossils from the Velkerri core, from the Velkerri Formation. And so this is just a very brief overview of methods. We macerated samples, we washed them with DI, filtered. They were handpicked out of resulting residues in a laminar flow hood to reduce contamination, and then packaged in ashed silver cups and ashed glass vials for analysis at Syracuse University. They were also cleaned with a little eyelash to get rid of any particulate kerogen that was stuck to the fossils. All right, so here is our data. So on the right here, I'm just showing data from Jarrett and Cox and others, just compiled molybdenum and TOC, just to give you that framework for the sort of 3 zones, sort of redox zones in the Velkerri. And here is our carbon isotope data. So the blue line is our bulk rock organic carbon data. Each gray dot is an individual measured fossil, layospheres. They're smooth-walled fossils, acritarchs. And then the red dots are the average for each horizon. And then things to sort of notice is that the mid to upper Waigori, the delta C13 of fossils, is heavier, lighter, and equivalent to the bulk, but the mean is overall generally enriched. Whereas in the middle section there where we have samples, the delta C13 of fossils is always heavier than bulk. And there are no fossils after this horizon. So these tick marks over here represent all of our sampled horizons. Okay, and then the middle panel here, Chris also ran kerogen and bulk nitrogen to sort of give us a little bit more information about what's happening in the water column here. And these slightly positive values are a hallmark of Mesoproterozoic strata and point to a redox active nitrogen cycle, cycling between oxic surface waters and anoxic deep waters. Another thing to note is that there's no relationship between fossil size and isotopic value. So we have delta C13 on the x-axis and diameter in microns of the fossils on the y-axis. This also gives you a sense of the size range of the fossils that we were looking at. All right, so here's our cartoon version of the interpretation of our data. That in the upper part of the section here, we have organisms living in maybe a thicker oxygenated layer, and therefore recording more variability depending on potentially the thickness of that oxic layer, as well as the efficiency of the biological pump, recording isotopic values that are going to be equivalent, heavier, or lighter than the bulk. Whereas in the areas of the core where we have independent redox proxy evidence for anoxia and euxinia at depth, we see that the fossils are always heavier than the bulk, which indicates to us that they're living in this maybe relatively thin oxic layer at the surface and recording those more enriched values from a steeper gradient and a more efficient biological pump. So if we come back to our 2 model scenarios, our data for this material supports this prevailing model where early eukaryotes were restricted to oxic habitats. So another interesting thing about this is that this yellow star here from the protosterol data are actually 4 samples from the top of the Velkerri Formation. And so there are protosterols in this that have been found in this core. Super cool coincidence. So this confirms that eukaryotes were living in oxygenated environments and supports the hypothesis that these protosterols are derived from eukaryotes as opposed to bacteria, which is another possibility. It doesn't negate that possibility, but it does add an interesting wrinkle to it. So if these fossils and biomarkers represent stem group eukaryotes that were living in oxygenated habitats, it does indicate that they had already required an oxygen-dependent mitochondrion. They were certainly oxygen-tolerant lineages. It doesn't tell us if they were in the official crown or not, but I think it does give us important information that we didn't have before about what Mesoproterozoic acritarchs, at least in this basin, were doing. And this is also a deeper water basin. This is not super shallow. And so that's another interesting thing to think about in terms of our ideas about where eukaryotes were living. There is more fossil diversity and abundance in other parts of the Roper Group that are nearer shore, and that's another interesting part of the story. There are many questions that remain regarding protosterols, the rise of crown group eukaryotes, and the subsequent eukaryotic diversification. But I think this is a really neat addition to our understanding of how eukaryotic ecosystems were structured in the Mesoproterozoic. Okay, so I'm going to move on now to use case number 2, Cryogenian-Ediacaran algae. So this is work that's much more preliminary. As you'll see, we're still gathering data on this, but another use of this nano-EA technique that we're really excited about. So this is material from the Tsagaan terrain of Mongolia. These were samples that were collected by Ross Anderson and colleagues. And much of the sort of geology, structural geology, sedimentology, and stratigraphy of this that forms the basis for much of what I'm doing was done by Francis McDonald and Nyanga Bold. So this is a figure, couple of figures from Bold 2016. So we have material from both the Kongor and Taishir areas, and in this little figure here, the purple with the yellow are the diamectites. So the lower one's the sturt, and then the top one is the marinoan. And there's 3 members through here, T1, T2, T3, indicated by the colors. And the little black dots here are carbon isotopes. So we have this Taishir isotopic anomaly in this sort of boundary between the T2 and T3. And previous work had found fossils in these samples, which I'll talk about a little bit moving forward, to give you the stratigraphic context here. So we're looking at the interglacial sediments. So there's been a lot of work done on these strata by many colleagues over the years. And so the 2 that are probably the most relevant for us today is this figure on the left by Dave Johnson and colleagues from 2013, showing these very tightly coupled organic and inorganic records through the Taishir. So the red dots are carbonate and the blue dots are organic carbon. And so they used these just to sort of make some interesting conclusions about the primary nature of this anomaly that would have required an influx of light organic carbon into the system. And then Tanya Bosak and colleagues started finding these really interesting fossils in these same rocks. And they originally interpreted them as ciliates. When I was a postdoc at MIT, I got some of this material from Tanya and started working on it because other interesting fossils had also been found in these same strata. These things that we decided to call MOWs, because why not come up with more silly acronyms, macroscopic organic warty sheets. So these are quite large. The scale bar here in this first picture is 1 millimeter. These are really macroscopic, you can see them. And they're organic carbon dissolved out of organic rich limestone rocks with acetic acid. And they are covered with these really interesting warts. So the top panel here is an SEM image. The lower left is a light image, so you can see these sort of darker warts over the course of the body of this fossil. And so in 2017, we interpreted these as being putative red algae. And in the bottom right panel is just a figure from that paper where we show some examples of modern red algae that have morphological similarity, including these warts, which in modern red algae are reproductive structures, where essentially the baby red algae are produced and then expelled from the tops of the warts. And many red algae have a crustose form. These are not biomineralized. These aren't like coralline red algae. A crustose form that has sort of a bumpy top and then a smoother bottom, which is also consistent with what we saw in these Mongolian material. So we sort of had these 2 different types of fossils. We had these little putative ciliates, and then we had these MOWs. And I had a student, Maoli, who started working on the putative ciliates, which we decided to call organic spore-like microfossils, because again, acronyms. And one day I was like, why don't we cut them open? So we cut them open and started looking at them under the microscope and discovered that they are not hollow, which is what you would expect from a ciliate lorica, a tintinid lorica, which was the original interpretation. They were actually full all the way through with cellular structures. And so this really changed our interpretation, and it especially changed our interpretation because we also did sectioning through those mouse warts. And so what we discovered is that they were the same. So if you look at this upper panel here, on the left, A and B are cross sections through warts on the surface of one of these sheets. C and D are cross sections through these individual spores. And so the size and morphology of these are statistically indistinguishable. E is an example of a red algal carpospore, so essentially a juvenile red algae. So a lot of morphological similarity, although the sizes are a little bit different. So our idea is that essentially the warts are producing these spores and that they are not ciliates. The lower left panel here, D, or the lower right, is showing what a sort of cross-section of a ciliate would look like. You might have a bumpy texture on the lorica, but it would not be solid cellular material all the way through. It would host a single-celled organism. So there's an interesting taphonomy story here as well, in that we never see the sheets and the spores in the same sample. And so we have some interesting ideas about why it might be that the spores are being preserved differently than the sheets. And that could be due to various resistance, and also the fact that spores are ejected only sometimes during reproductive cycles. Okay, so the fact that they're red algae totally makes sense because we already have Crown group red algae 1 billion years ago. And so there's really no inconsistency here. And it's really amazing that we're finding them in between these two snowball Earth events. And so because they're made of organic carbon, this had been shown by Tanya and then again by us, we decided to start analyzing them using the same techniques that I told you about before. So we use the nano-EA to measure fossils, and then we also did our carbonate carbon isotopes through this interval. And so our results are over here on the left. So we have from the Taishir area, we have T1, T2, and T3, although we don't have any fossils from T1 here. And then in the Kongor range, this is like a compiled strat section just to keep everything a little bit easier to see. We have OSMs from the Ediacaran. And that's really cool too, because that hadn't been seen before. And so basically what we're able to show here is that this lineage was persisting through the Marinoan glaciation in some form. The carbonate carbon is in light blue on the right. And then on this right panel here, I'm just showing you some box plots of the isotopic value of the fossils by member. And the point of this is to just show you that they're incredibly consistent and they don't vary very much. There is more variability in the Ediacaran material than there is in the Cryogenian material. And it's very light, okay? So 31 to 32 per mil, which is quite light. Okay, so at first we were a little bit worried about our data being quite so light. But then we started doing some more digging and found something very, very cool, which is that there are a bunch of modern phycologists who have observed that modern red algae have very light organic carbon isotopes, and no one really understands why. So this is a figure from a Raven paper that compiled a bunch of data on red, green, and brown algae from England, Scotland, and Norway. And basically what they're showing is the number of species with delta C13 values that are less than -30. In the rhodophytes is 21. It's 0 in the brown and green algae. So there's something unique about red algae that are leading to this more depleted carbon isotopic signature, which is something that we're reading and learning more about, trying to understand. But this was really cool because it means that the carbon isotopic signature is consistent with a red algal affinity. Okay, so what else does this mean? We also have this really wonderful paired data from Dave and colleagues, and they present some interesting ideas about the source of this pairing, but you see this really strong covariation between carbonate and organic carbon. So if we look at our cap delta, so our offset between our carbonate carbon and our organic carbon, we see something which I think is pretty interesting, which is that there's a big shift here. And the big shift is happening because the fossils are not tracking variation in carbonate delta C13. The carbonate is changing. If we go back, you can see that the carbonate here is getting lighter, but the fossils are just chugging along at around -31, -32, doing their thing. And so when we look at this in sort of cap delta space, we see this shift between what's happening in the Taishir and then what's happening up in the Ol. This dotted box is the approximate cap delta 13C of bulk organic and bulk carb from Dave's paper, so the offset there, which is also really interesting to me because it seems like the Ol is showing consistent offset, whereas the Taishir, the fossils are recording a different offset than the bulk. Unfortunately, we don't have our own bulk organic data yet to compare to, which is obviously something that we're working on. But the fossils are staying the same, and it's the carbonate that's moving, which is something that we're still trying to think about and understand. So we're going to be running our own bulk. The other thing that you'll notice maybe is that we actually don't have any fossils through the anomaly itself, which is essentially right here in between T2 and T3. And that's just an unfortunate fact of the sample suite that we had access to. And so hopefully I will be getting access to more samples that will cover that range, because what would be really cool is to see what's happening actually during the negative anomaly. Are the fossils just chugging along, and are they unimpacted by it, or are they tracking DIC when variation is extreme? And that's all I've got for today. I know that was kind of a fast talk, but hopefully that leaves us lots of time for questions. So 2 use cases of this single fossil carbon isotope data, one from the Mesoproterozoic and one from the Cryogenian-Ediacaran.
Okay, really cool stuff, Phoebe. Thanks a bunch for that. So now we'll go ahead and switch to our questions and discussion. So there's a few ways that people can ask questions. They can find the raise hand button by clicking react at the bottom of the screen and clicking raise hand. Or you can type it into the chat if you would like us to read it out for you. So first hand up was Adriana Rizzo. Please unmute yourself.
Hi, Phoebe. Great talk. Very interesting data set. I have a lot of questions. But I think my first question, most of them are about the Velkerri work, I guess. So with that data set, it seemed, maybe I missed something, but you were measuring layospheroids, correct? I guess.
Yes.
What do you know about where they live in terms of depth? Is there any way of determining whether they're surface or benthic?
Well, yeah, I mean, we would argue that our data supports that they were living in the surface, especially in the middle member where we're seeing very depleted values. It is possible that they were benthic. The problem with acritarchs is that we don't exactly know where they're living, but many of them are assumed to be the cysts of photosynthetic algae. It is also possible that some of them represent heterotrophs. But the fact that they are recording such heavy values in a basin where we know we have a steep carbon isotopic gradient, to us confirms the fact that they were living in the surface ocean.
Okay.
Doesn't necessarily mean they were photosynthetic, but that's where they were incorporating their carbon.
Okay. Sorry. I'll ask my other questions later to give some other people a chance.
Thanks, Adriana. Okay, Greg, please go ahead next.
Yeah, that was really great, and I really love those. I really love those cryogenian fossils. But I wonder, in the terms of eukaryotes, have you thought at all about that wonderful fossil that Stefan Bengtson described called Myxomatodes, which he regards as a slime mold, which of course is a eukaryote because it forms an actual slug that can move along and leave something that looks like a trace fossil. I've worked on them as well, and there's also another thing called Discagma. These are both 2 billion years old. And as an aside, I'm sorry to announce that Stefan died just 2 days ago. And I'm wondering if anyone knows what the cause of death was. He was a good friend.
I'm so sorry to hear that. I didn't know that. Thank you for
Yeah.
sharing that sad news with us.
Maybe someone knows on this feed. But anyway, what about these 2 billion-year-old possible eukaryotes, which also are in oxidizing environments?
I don't think they're eukaryotes. I think they're interesting, but I don't think that there's enough compelling evidence that they have a eukaryotic affinity. I don't think that the threshold of certainty has been met, and it's inconsistent with many other lines of evidence that I've presented here in terms of thinking about phylogenies and things like that. So I think they're interesting. I could be wrong. I don't think they're eukaryotes.
Well, that's fair enough. They're right on the great oxidation event, of course, which feeds to your point about oxygen being important.
Yeah.
All right. Thanks, Greg. Paul, you have your hand up next.
Yeah, regarding the Mesoproterozoic Velkerri material, incidentally, very fascinating results. I'm wondering whether the variability in the carbon isotopes, rather than reflecting different water depths in a sort of steady state, might instead reflect production during algal blooms. You might expect that during blooms, there would be a progression toward more depleted values.
Yeah, that's a great point, Paul. And yeah, in the paper on this, we do talk more about the spread within an individual sample. And there are so many things that can influence that. If you go into a modern environment and do this, like if you measure, there's been some really cool studies that have done this with foraminifera, as well as with Daphnia epiphyia, which are like little Daphnia eggs. Both in the modern and in the more recent fossil record, you do see a spread of up to 10 per mil in basically non-time averaged material, which is really fascinating. And so because the lifespan of an individual organism is so short, those sorts of variabilities that you're talking about could get recorded in an individual fossil on like a seasonal basis, right? And we think that's partially responsible for the widespread that we are seeing like within an individual sample, which is of course time averaged itself.
Yeah, well, I gather blooms have a large number of potential causes, often driven by viruses. Yeah.
All right. Thanks, Paul. Ava, you are ready next. Ava Stoican, please go ahead.
Oh yeah, thanks for your talk. So I was wondering if you use mass balance to calculate essentially the fraction of surface eukaryotic biomass and the fraction of deep water, I guess, prokaryotic biomass, if that's similar to modern, or if that would tell you something maybe also about these algal blooms potentially, if that would be a useful
definitely. We would love to do some mass balance. We haven't gotten there yet, but it's sort of on the list of things to do. One of the problems with doing mass balance here, and Hedda has done some complementary work that we are hopefully publishing soon, is that we only get to look at what's been preserved. And so in terms of the like physical fossils, whereas the bulk is probably recording stuff that we can't physically identify. And so we could do it. It's just a little bit hand wavy because we can't quite like get at the whole story, but agree that it's an important component of this that we need to do. Yeah. And could be really interesting in terms of thinking about what's pushing the bulk one way or the other.
Yeah, right. Well, thank you.
All right. Thanks, Ava. Shuhai, please go ahead. Shuhai Zhao.
Yeah, hi, Phoebe. Great talk. Thank you. Thank you very much. Can you hear me?
Yep.
Okay. Yeah, I was intrigued by the comment of the red alga interpretation and the isotopes. So this is a diagram I would like to comment on. So in the Mongolia specimen, you got -32, and you said it's consistent with, almost uniquely consistent with red algae, because I want to also get your comment on when the uniqueness of this light carbon isotope. It is unique to red algae, or it is also available, consistent with the other algal interpretation. But my comment here is if you look at the your Australian specimen, the acritarchs are also very light, right? So it's lighter in some cases. So if the isotope is a uniquely red algal, minus 30, 32 or lower, then you would take this argument and say, all these acritarchs from Australia are also red algal, right?
Well, I see your point, but I think there's a couple differences here. One is that the red algal data is extremely consistent throughout many hundreds of meters of section. And that's not the case with the eukaryotes from the Velkerri. We have means that are, negative 27 or even negative 25 in the Amungi member. And as I said to Paul, the range that we see in an individual sample could be due to many different factors. And so we can't, having one sample that's at negative 35 does not mean that isn't a red algal in my interpretation. We have to take a statistical argument here.
So
the other thing is that the red algae are much more these are multicellular organisms that probably are capable of much more complicated, photosynthetic carbon sequestration, processes that would potentially impart a unique and stable carbon isotopic signature, which, a Mesoproterozoic stem group eukaryote might not be capable of doing. So your point is well taken that how can I say one is due to taxonomic variability and the other is due to water column dynamics? And, I think the red algal interpretation or the use of isotopes to derive the red algal interpretation is something that we're still very much investigating. And so I presented here as sort of a tantalizing idea based on our understanding of modern red algal isotopic signatures.
Yeah, okay, great. If I can, I have another comment on the, just the variation of isotopes on the left panel here. Just looking at 10 per mil or greater isotopic variation, and particularly if the lower part, the acritarchs are almost always greater than the TLC. So that means that there's some extremely light component in your TLC that is probably minus 38 to minus 40 per mil in order to balance the mass.
Yeah.
So I don't know how to explain that. If you have water column isotopic gradient of greater than 5 per mil, it's difficult to sustain this, right? You're thinking about you're sending all this organic carbon to the deep ocean. You're also sending the nutrients, the phosphorus, the nitrogen to the deep ocean, and to sustain a 5 per mil, 10 per mil depth gradient in the DIC, it's difficult because sooner or later you're going to exhaust the nutrients in the surface ocean. So the pump is not loses steam eventually. So you cannot sustain a very strong pump to over what is the residence time of the carbon, over a minute year.
Yeah.
It's difficult to do. So I wonder whether you have any thoughts about this huge gradient here, a huge variation that you, particularly in the lower part, probably 15 per mil.
Yeah, no, it's a good question. I think I think the cartoons that we present, the visual cartoons that we present, are very much cartoons, and I don't think that we're expecting that there would be a stable, very steep gradient for hundreds of thousands of years or anything for the reasons that you explain. But we think it is possible that gradient could be steeper, right? That there could be enough of a difference between oxygenated and non-oxygenated time periods in the Velkerri, which could be due to changes in water depth or nutrient input, that it could get recorded in these organisms.
Okay.
But your point is well taken. Yeah.
Well, thank you very much. Great talk, Phoebe. Great.
All right. The next question was in the chat from Emmy Smith, which I'll read out loud. Emmy says, "Very interesting work. Thank you for presenting. Sorry if I missed this, but would you be willing to speculate on why you think the MOWSs are only found in the OI and OSMs, are only
Like why they don't
in the
co-occur?
Tashir?
Why we don't see MOWs in the OL? Is that maybe what the question is?
It seems like it. I'll just finish the last.
And the OSMs are only found in the Tashir.
Yes. So we find MOWs in the Tashir as well, but you're right, we don't find them in the OL. I have no idea. It's a great question. I think the MOWs are in general are much rarer and at much lower abundance through the sections that I've been able to look at. And we're a little bit sample limited on some of this stuff. And one of our ideas is essentially that there's parts of the life cycle of the MOWs that have higher and lower preservation potential, whereas the OSMs have more consistent preservation potential. And it could, it's a very unsatisfactory answer, but it could be a fluke. But I don't think that I've spent enough time thinking about it or looking at, say, differences in clay chemistry or other things that might have taphonomic control. But it does put a chink in the idea that they are coming from the same organism. But I do find the cross-section microscopic analyses very compelling, as well as the paper has like size distribution statistics that showing that they're the same size. So I think it comes down to parsimony on that one. Yeah.
Thanks.
All right. Adriana, I had another couple questions. Go ahead.
Yeah, thank you. Just kind of following up on the question. So the questions about the previous question and some of the questions about the isotope gradient. I guess I'm wondering, do you have evidence for the DIC gradient in the Velkerri beyond this the Laosphere carbon isotope measurements? Because do you have independent evidence for what, like, any idea of what?
I mean, I guess the modeling combined with the redox and TOC data would be my answer. Yeah, I'm not quite sure what else we would be able to access. I'm happy to take ideas though, if you have one.
Yeah, I mean that's why I was asking about the benthic versus surface, because if you had some sort of independent constraint about what habitat they're living in.
Yes, I mean that is one of the questions. That is essentially the question we were trying to get at with this. We don't have any independent constraints on what habitats they're living in, which is part of the reason why it's hard to interpret this idea about where organisms were living in the Mesoproterozoic.
Yeah. Okay. And then my other question was, you may have said this, but I missed it. But in Brox's work they found in the 2023 paper, they found these like sterols, as they call them, with the 24-methyl protosterols. Do you know where stratigraphically those samples come from relative to your data?
Yeah, so it's from 4 samples in the Waiwuri.
Okay.
I can't remember exactly which ones, but I believe it's near the top.
Okay, but it's same. Yeah. Okay.
Oh yes, it's from the same core, amazingly.
Yeah. Okay.
So yeah, four of them. Yeah. Mm-hmm.
Okay, cool. Thank you.
Yeah, but they did not, as far as I know, they did not look in the Amunji or the Kalala because probably they didn't think they would find them there. The thermal maturity also increases down core. So we don't know if they're present in other parts of the core. They were only examined in that top member. Right. And there other folks have done previous biomarker work that show that there's no sort of regular steranes in this core.
Yes. Yeah.
Yeah.
Okay. All right. Thank you.
Yeah.
Appreciate it.
All right. Thanks, Adriana. Andre, please go ahead. You're muted.
Sorry. I'm just sort of commenting on the same question that everyone asked you about the stratification. So you're analyzing all your fossils are eukaryotes and maybe the same species. So this way, I guess you can not the same species. So
I mean, we have no way of knowing. They're the same morphospecies.
Morphospecies.
Yeah.
So at least to some degree, you exclude influence of metabolism.
Yeah, exactly.
To a degree you can. So when sort of there is a possibility that environmental factors control this range, like Paul mentioned, like algal bloom, which would result in more heavy values. And your heaviest values are not super heavy. For algal bloom, but I mean, maybe. But I'm wondering if you now have two options, environmental control versus stratification. Can you actually say that was dissolved in organic carbon stratification in your basin? And what does it mean for your implication that eukaryotes lived below and above redox climate? And I thought one way you could potentially answer it, if you would take the sample, like very thin sample, and extract multiple samples from it and analyze them. So how much of variability would you get if you pick up, because this acritarchs probably will live very short. And if you find them in a very small sample, if you take them from the same sample, probably they represent like maybe several years. So this way you limit environmental effect to some degree. Would you see still 10 per mil variation between them, or they would be much closer?
Yeah, I mean, these are extremely small samples that we're working with already. So we tried our best to do that already. And so the variation that you're seeing is probably as good as we're going to get in terms of time averaging. The fossils are not extremely abundant. So it wouldn't be possible for us to say, float them off of a single bedding plane. I don't think we would get enough. But as I've said, there's lots of work done in Paleozoic, eukaryotic marine microfossils, as well as in the modern, that shows that a 10 per mil variation can happen over the course of a year relatively easily because of things like algal blooms and other variations. So the range that we're seeing is very consistent with other published work, including our work in the Devonian on Paleozoic acritarchs, which also shows a similar range.
But if you sort of say that it's environmentally controlled, can you then say anything about stratification? Because kind of how do you...
Well, I guess we're looking at means here, right? Like the range and the mean versus compared to the bulk, like the cap delta value is different than the range. Like I think what we're interested in isn't the range, but the range in relationship to what the bulk is doing.
Okay.
Yeah.
All right. Thanks, Andre. Paul has a question next. Andre, I'm gonna exit and leave it to you, okay?
Okay.
Thanks, everyone.
Yeah, Phoebe, have you published the work on the internal structure of the warts and the pseudociliates from the Teichir?
Yeah, it's published in Paleontology.
Okay.
2017.
2017?
Mm-hmm.
Okay,
yeah.
Great, thanks.
Yeah, the isotopes, none of the isotope stuff is published yet, but the fossil morphology material is published yet.
Okay. Any more questions for Phoebe? Looks like most people satisfied. Okay, we don't have anything in chat. Nobody raising hand. Paul, do you have? Okay, Shuhei corrected it was paleontology 2020, and Adriana.
Oh, sorry. Yes, thank you. Shuhei. Can't remember the dates of my papers.
Okay.
I could ask another sort of pseudo question. Regarding the absence of the early steranes, it's always bothered me a little bit about the biomarker data that it comes almost exclusively from pyroxenes because of the requirement that it be unmetamorphosed.
Yeah.
So, and so pyroxenes are often prone to oligotrophy and thermal stress. And you might expect that the eukaryotic record would be richer on continental margins where there are upwellings, which are cooler and more nutrient rich. So I'm wondering whether that, I've often wondered whether that could be part of the explanation for the absence of the early steranes. I guess the counter argument is that when the steranes do come in after 800, it's from similar areas.
Yeah, I think that's a great point. I think there's a lot of ways to explain the sterane record that are consistent with the fossil record. I think that's one of them. And I think an argument to sort of make your two statements consistent would be, okay, some oxygen threshold was reached in that time period, right? Or a decline in euxinia, maybe in hyperic so that, you have some shift where suddenly eukaryotic populations can reach a point in those areas where they can get recorded in the sedimentary record, right? Because some of this has to do with like a total biomass in terms of being able to show up. So that's one idea, maybe, is that yes, there's these basinal differences early on, and then later those basinal differences are less important because of changing redox conditions in the ocean. I think taphonomy also probably likely plays a role, but yeah.
Seems to me the neo-oxygenation event occurred in the Devonian. Yeah,
like the deep water, right? But I think that, like, for animals, I think one of the tricky things is like how much oxygen does a single-celled eukaryote need, right? And it's not very much. And so it could be that there's thresholds that are being met along the way that like don't rise to the occasion of the evolution of metazoa, but are still important.
Yeah.
Maybe.
We could use more data from the modern on that.
Yeah, tell me about it.
Okay, so some of the hyperic were quite organic rich, like the one that you studied. But Adriana has a question. Adriana, are you still here? Do you want me to read it or you ask it?
I was going to ask if you knew anything about the composition of in terms of if anybody's done like ramen or some sort of analysis like that on the composition of the Velkerri acritarchs, perhaps in contrast to your cryogenian stuff, because I guess carbohydrates, are often complex carbohydrates are often hypothesized to be a major component of some organic world microfossils, and carbohydrates tend to be isotopically heavier compared to other types of organic matter. I wonder if you'd thought about that at all.
Yes, we have thought about that. And I don't have a good answer for you. I think we thought about this more in our Devonian paper, which is in geobiology a couple of years ago. There's been tons of Raman and FTIR done on various organisms. There is Raman data on the OSMs that was done in the original paper by Tanya and colleagues, by both second colleagues. We didn't do any additional Raman or FTIR. I don't know if anyone's done. Emmanuel Javot did, published some of that work on Roper fossils from lower down in the section, but not from the Velkerri. So that's an interesting question. Is there something in that data that would point to compositional differences? My interpretation of a lot of that data is that we don't know enough to really say. There is no clear signal that like, oh, these organisms have more carbohydrates than these other ones, and these ones have more lipids. But you're right that we do know from the modern that different components of an organism are going to have different isotopic signatures. Our take has always been that these are extremely recalcitrant structures that are not likely going to have, say, like a high protein signature, for example. And we go into a little bit more detail in our geobiology paper, and it's not coming to me right now, so I apologize for not having a slightly more nuanced answer for you. But yes, excellent point and something that we've thought about.
Okay,
Great, thank you.
Bruna Baker-Kerber asked question. Great talk, Phoebe. I have just one quick question. Is there already a paper on nano-EA setup? I think Chris Janum had a paper, maybe rapid communication and mass spectrometry. I don't remember, was it 2017 or something? But anyway, Bruno, if you contact Phoebe or me, we can send you a paper. Okay, and I guess next, Susanna.
Hi, Phoebe. Great job. Exciting talk. I had a question about the Velkerri results, the interpretation that it indicates that these eukaryotes had mitochondria, and I ask because I'm struggling with this myself. This question, I used to assume that if you found eukaryotic fossils in oxic conditions, it would indicate mitochondria. But now that there's evidence for Asgard, archaea that live in oxygenated environments and are aerobic, presumably, I don't know how that changes things. What do you think?
Yeah, that's a great question. And I guess the question is, do we think Asgard archaea type organisms would make resistant preservable structures? And I guess one argument would be, well, yes, because they're the cytoplasmic bag of the eukaryote. But I don't know that we have much data from the modern to support that. Maybe you know more than me. So I guess that would be one of my arguments. But your point is well taken. I think that I do not I think what we can say is that they're living in oxygenated environments. If they're eukaryotes, then that means that they had mitochondria, right? Like
if they were eukaryotes, then they probably had mitochondria.
Yeah, exactly. Yeah. I think I'm excited about this because it feels like incremental progress towards this problem, but I don't see it as like, okay, we've put a bow on it, you know? Yeah, but it's a great point.
Thank you. Paul, you have something?
My question is for you, Andre.
Okay.
I think you should change your background slide because if those are seagrasses, they're angiosperms, and we can't allow them in the Precambrian. I mean, unless you're trying to use them as a zone fossil for your own existence.
Okay. Yeah, I'll see if I can find something different. Okay. Any more questions for Phoebe? Okay, last call? No? Okay, thanks, Phoebe. I think everyone enjoyed it, and we look forward to talk next week by Kurt Konhauser. Thanks, everyone, and see you next week. Bye.
Thanks, Phoebe.
Thank you.
Bye.